Thank you, Manos. Thank you, Rebecca and Areti, for your great talks. I'd like to take this opportunity to congratulate Claudia and the entire TAB team for a really impressive Biennale. And I'm very pleased to be here in your beautiful city of Tallinn. I don't see anything on screen. Do you see something on screen? No, we're just waiting for my presentation. And then we'll get warmed up. So whilst that's loading, the title of this symposium, Polycephalum City, is literally translated as the many-headed city. And the inspiration for exploring the urban landscape as this multiplicitous being is the slime mold, Physarum polycephalum. How are we doing? If you want to stretch at this point, you have been sitting for an hour, then take this opportunity to stand and have a little stretch. Have we? Hey, there we go. Okay. So, as I said, the slime mold is the inspiration for the title of the symposium. Thank you. Since 2009, I have been working with this species of slime mold, Physarum polycephalum, as an artist, a researcher, and an educator, developing a range of studies, methods, practices, employing the slime mold as artistic medium, educational model, and social metaphor. And today, I would like to share with you a few reasons why Physarum polycephalum is an important model organism in biological, technological, and social terms, and why it's such a fitting namesake for this many-headed symposium. So, this is Physarum polycephalum, and thank you, Areti, for not saying too much about slime molds, though obviously you have been introduced through the 2 days of the symposium. So this is a yellow cellular mass which grows in branching forms. It's a single-celled organism, so which means single cells join together with others of its own kind to form a mass supercell. So within a slime mold cell, you might have thousands, sometimes millions of individual nuclei, all working together, all operating as one single entity. In its natural habitat, it can be found in temperate woodland, feeding on rotting vegetation. But you can just as commonly find it in the laboratories of biologists and roboticists and the studios of artists and architects. Why? Because this bag of cellular slime can perform high levels of computation and demonstrates highly efficient communication and cooperation strategies. All of this without any sensory organs, no central nervous system, and no brain. It's a curious nomad. It's highly exploratory, and it's impossible to domesticate. It's untamable. Here it is escaping from one petri dish into another in my studio. I began working with slime mold some years ago when a microbiologist friend of mine gave me a little petri dish on a lab visit with a yellow blob in it and invited me to go home and play with it. The only instructions I were given was that it likes it dark and damp, and its favorite food was porridge oats. I began observing its behavior through time-lapse photography, essentially. Its top speed of growth is 1 centimeter an hour, so it's not something that you can view particularly in real time. But through time-lapse photography, interesting behaviors revealed themselves. So, for example, here, having fed on a pile of oats, the slime mold goes off exploring in 2 directions simultaneously. When it approaches itself, observe what it does. It pauses, it retreats, and it pushes its energy into another direction. It redirects its growth. So this organism possesses both temporal and spatial memory. As it explores its environment, it leaves a membrane trace behind it, indicating where it's already been. And this is only one of its very clever tricks. So this residual trace operates like an extended body, a mechanism for leaving information in its environment, a stigmergic signal for itself and others to find. Stigmergy being the biological process where an organism leaves a physical trace in its environment, which then affects the behavior of other organisms. So think of ants leaving pheromone trails, or termites leaving mud balls to indicate what needs to be done. Or in human terms, we could think of how graffiti encourages more graffiti, or rubbish being left encourages more rubbish to be left. We develop patterns from observation. So the slime mold can learn, it can problem solve, it can navigate efficiently, and it can optimize networks, all without any form of centralized control. So the question is, how is it able to do this? How is it able to perform tasks that we associate with brain function? For this, let's take a closer look. So this is a microscopic video that I shot. It's 100 times magnification and sped up 20 times. So as it explores its environment, the slime mold pulses in a rhythmic flow. It's a streaming motion, shuttling back and forth within the mast cell. So it's transmitting information about its environment and nutrients first in one direction and then back in the other, each pulse lasting a minute or two. And whilst no one knows exactly how it's computing and operating, it is within this continuous synchronous oscillation that researchers believe the intelligence of this organism lies. So the transmission of these chemical signals giving information, distributing information across the global property of the organism, communicating vital information about its environment and transmitting resources through the body cell. Within this unicellular structure, all parts of the organism work together as one. They're maximizing resources and opportunity. There's no top-down control, only local interactions and a constant communication loop within this cell structure. In ecological terms, the slime mold has little use. If all the slime molds in the world, and there are about 700 known species of them, if they all disappeared, we might not notice ecologically. But as a model organism, it's a different story. A great many fields of research are asking different questions of this organism to better understand how something with such simple elements can complete complex tasks. There are over 50,000 published academic papers on slime molds, over 20,000 on Physarum polycephalum alone, the species that I work mostly with. And to give you a flavor of that research, one study, scientists at Hokkaido University, some of you may be familiar with this, filled a small maze with a slime mold. It joined together and fused and formed a mass single cell. They introduced food at 2 points, oats, of course, and the slime mold retreated from the empty areas of the cell and formed a strong line of connection connecting the two food sources. Now, there are 4 possible routes through this maze, but time and time again, the slime mold found the shortest and the most efficient route. And the conclusion from this experiment was that the slime mold demonstrated a primitive form of intelligence. Another experiment, which some may be familiar with. So researchers invited the slime mold to explore a territory scattered with oats. It branched out from the center to explore the territory, locking onto each food node as it went, forming connections and over time establishing a network between all the food nodes. That's not unusual, that's what the slime mold does. Where this becomes interesting is the central large node represents the city of Tokyo, and each surrounding food node represents an outlying suburban railway station. So the slime mold had successfully replicated the Tokyo transport network, a hugely complex system. So what took us, human beings, well over a century to establish through community dwelling, urban planning, and civil engineering, took the slime mold just 26 hours. And since this demonstration, slime mold has mapped the world, including UK motorways, US highways, migration routes, drug trafficking routes, evacuation routes. If you want to lose a few hours on YouTube, just type slime mold map, and you can travel the world through the slime mold's navigational abilities. So the world over, researchers are decoding its biological behaviors to extract the computational rules and then apply those in very diverse fields of inquiry. Just a couple of examples. On the left, you have a computer model by Jeff Jones, and this is simulating the Tokyo experiment. So this is an algorithm derived from the biological rules. And on the right, we have a robot controlled by the slime mold. We'll just enjoy watching the robot for a moment and watch the network rationalize in the most efficient way. Other investigations include musical composition, management studies, philosophy, and numerous urban design applications exploring swarm behavior, resource distribution, and network optimization. There are lots of people working with the slime mold. If you don't wish to read all 50,000 academic papers on the subject, you might instead want to watch the award-winning feature-length documentary film, The Creeping Garden, which tells the story of slime mold behavior and experimentation through the people that conduct diverse disciplinary inquiry with the organism. So for me, as an artist, the slime mold is my working material. It is a living co-producer in the creation of films and prints and photographs. It's a fascinating subject, biologically and computationally. And I often work in collaboration with scientists or take scientific research as a stimulus for my works. This is my homage to the maze experiment. So I built a large replica of the maze and invited the slime mold to freely roam, making decisions on which turn to take in search of food. And over time, I've grown to understand its motivations and its behaviors. I can predict its patterns of growth. I know its likes and its dislikes. I know how to keep it happy, how to farm it well. But ultimately, I cannot control the outcome. And the aim of my work is not to illustrate scientific principles or to represent nature, but to draw attention to the aesthetic and behavioral qualities inherent within such simple yet complex biological systems, to enable the organism to reveal itself through my interventions, slime mold as co-creator. And rather than trying to predefine the outcome, I want to create the conditions within which interesting behaviors can emerge. I also work with computational models of the organism. Here, an interactive piece for exhibition. And this is where visitors to the gallery are picked up through motion capture and then transmitted to the screen as virtual food nodes. So they become oats for the digital slime mold to consume and to network between. The slime mold simulation finds the oats and forms networks between them. And as people move in the space, they alter the formation of the nodes and subsequently the networks that appear on screen. So here you see people trying to draw with the virtual slime mold. So slowly drawing with a digital organism and also a virtual kind of hand-holding for the visitors to the gallery. And the digital simulation responds to this ever-changing network, adapting to the changing growth patterns. My contribution to the exhibition here, Anthropocene Island, was produced in collaboration with Ecologic Studio. It was an invitation for the biological computer of the slime mold to navigate the reimagined topography that they modeled of the peninsula Palisari. So I'll show you some excerpts, and you can see the full film in the exhibition if you haven't already. So here, as individual slime mold cells traverse the informational landscape in search of nutrients, and as they meet, they fuse together to build a critical mass. So what starts off as individual cells becomes united as one. This improves their chance of survival through collective cooperation. So you can see it joining together, networking as it goes. If we take a closer look, pulsing and flowing, the slime mold contours across the landscape. And if you look at its growth behavior, when it's searching for food, it branches out in multiple directions to maximize the amount of terrain it can cover. When it locates food or resources start to deplete, it will rationalize its growth to form this tubular network, adapting its behavior and its morphology in response to environmental change. So here you can see the rhythmic oscillations pulsing as chemical signals are transmitted across the entire cellular structure, a living information distribution system. This is a tale of adaptive resilience, of biological strategizing in a changing world. So the modelling of slime mould behaviour in my work extends beyond manipulating biological material and computational modelling. I'm also interested in its metaphorical properties, which provide rich territory for exploring wider networked and collective behaviour. So the slime mould and other biological systems providing a stimulus for creative investigations into the complex interrelationships between biological, social and technological systems. In recent years, I've developed a number of collective experiments as a form of bio-urban modelling, which transcends species and transcends scale. And I have a couple of examples to share with you today. So in 2005, this was in association with a Physarum Transport Network workshop, so a specialist slime mold science conference held at Columbia University in New York. As an aside or complementary project to the conference, I created Nodes and Networks. And this was a platform for interdisciplinary inquiry and experimentation, comparing the behavior of slime mold to that of the city. So Nodes and Networks had two key phases. The first was an invitation to a group of professionals from different disciplines interested in biology, urbanism, network systems, bring them together to interact, to exchange ideas and expertise, and to devise experiments. So this is a group of ecologists, artists, behavioral scientists, urban designers, games designers, city planners, thinking of ideas of experiments that we could run with the public. The second phase was to then invite a public audience to come and share in that co-creation and experimentation. And the experiments that we devised took place across New York in laboratory, in the park, in the gallery, so situated within the city itself. We worked with slime mold as material in the lab, so you can see agar casts of the New York boroughs, which then participants used as a model to create their own cartography, using attractants and repellents to symbolize social factors in that environment. So, for example, they would explore gentrification or pollution or economic situations of that borough through mapping out with attractants and repellents that the slime mold would respond to. Top right, we modelled its behaviour through direct action in Central Park. So this is a behavioural experiment of humans following slime mould rules to test their capacity for cooperation and communication against that of the single-celled organism. Usually when I do this experiment, humans are not as good at cooperation and communication as that of the slime mould. We're too complex and ego tends to get in the way. We tracked human patterns of movement by observing people in the Metropolitan Museum of Art, a form of culture foraging. Whilst back in the lab, slime mold navigated a 3D printed model of the same galleries we'd explored. So whilst we were exploring how humans operate within the space, slime mold was operating, exploring a model. So the many-headed slime mold here was our inspiration, but its properties also emulated the way in which we were working. Different people with different ways of working, different ways of thinking, working together, bringing those different expertise together to conduct exploratory and inclusive experimentation. Another project more recently in London, which was inspired by bio-urban inquiry, happened in July of this year. And this is where a team of researchers and practitioners came together to embark upon a month-long situated residency. And we're based in our Bike Gallery in Hackney Wick. Hackney Wick is an area of London which has undergone significant economic and environmental change in recent years. It's an old residential area. It has the highest population density of artists anywhere in Europe. And it suffered, or experienced, should I say, the imposition of the Olympic Park, which London hosted in 2012. So there's been a lot of upheaval and a lot of different communities and groups vying for territory. So we connected visual, digital, and performance art practices with contemporary scientific research, law, and urban design. And the project Crowd Control explored the mechanisms of collective behavior through observation, simulation, and experimentation. So what we did was devise games, activities, experiences, and experiments to observe, test, and play with the control mechanisms that affect how individuals operate within groups and how they change to responding environmental conditions. So again, the experimentation here is not predefined, but emerged from the multiple interactions between the disparate individuals. Points of reference shifted across scale and between species, so from the collective communication of single-celled organisms to group dynamics of social animals to human demographics and changing populations. The question of control was explored at 3 levels. We were interested in that within the body, so the internal mechanisms that affect our behavior, such as stress response or personality type. Control between bodies, the interactions between individuals, such as social cues, sensory communication, the unwritten rules that we respond to. And beyond bodies, so looking at the external factors that set out to permit, persuade, or prohibit behaviors such as urban infrastructure, the materiality of space, legal norms, or technological interventions. And through studying group dynamics across different scales, crowd control aimed to observe and understand how groups move together, how they make decisions, how they transmit information, how they respond to their environment, and emergent exploration of emergent systems across scales at a human level. So with both these projects, the focus was on bringing people from different disciplines together, from academic and cultural practices, to conduct situated experimentation, which was responsive, and to engage a wide public in that process of co-inquiry. The nature of experimentation prioritized experiential and exploratory methods, with much activity taking place within the public realm. So we worked in the streets, the parks, the canalways, galleries, shopping centres, etc. So a form of active research which combines critical and creative approaches. The final project that I want to share with you today takes experiential research a little bit further. In recent months, I've been collaborating with a Swedish choreographer, Emma Ribbing, exploring dance as a research tool in urban planning and regeneration. The location for this research is a former railway factory in Malmö, Sweden. This industrial heritage site has been preserved largely through the cultural activities of its more recent inhabitants, the creative industries and the artist studios, which have transformed the abandoned industrial buildings into the cultural hub of Lokstallama studios. So Spatial Negotiations is a sensory exploration of urban space, inspired by the single-celled organism. So using as a starting point the collective behavior and spatial awareness of Physarum polycephalum, a group of dancers were invited to explore the historical architectural site, both inside and out. The aim of the project is to use dance or movement more generally as an experiential and sensory research tool, to elicit an embodied exploration of space, responding to environmental cues and materials. The outcomes of the collaboration so far include a short film, and if you want to play the film, I can talk over that. I don't think this one starts automatically. Thank you. So the film, which will be screened in full at the gala tonight, and it follows the dancers as they experience the materiality and the atmosphere of the urban space, whilst attempting to operate as a non-human collective body. In addition to having its own inherent aesthetic properties, the film is also intended as a consultation tool, the spatial negotiations of slime mold and/or humans as a stimulus for discussion with urban planners and developers, to encourage them to consider the interconnections between biological, human, and urban systems when they are developing their planning strategies. So in Malmö, conversations are planned to take place in the coming months between Emma, the choreographer, and local urban planners. And we'll see if this many-headed approach has any influence or impact on those development conversations. At the heart of all my practice is the bringing together of diverse methodologies that harness curiosity about how our world works across scales and systems, to cross-pollinate ideas about how we might ask those questions and ask questions of the world, and encouraging self-discovery in others through interaction and active research. These principles can be applied to how I work with living materials, with collaborators on a collective experiment, and with public participants in co-creation. In the different contexts in which I work, biological systems provide a platform for raising questions about collective intelligence, social organization, and individual agency, amongst many other things. And rather than trying to predefine or control the outcomes, I focus on creating the conditions for interesting behaviors to reveal themselves through processes of interaction, pattern recognition, and negotiation. So it's an emergent model for experimentation and one that continues to evolve. Thank you for your time.